Hi, good morning, everyone. This is Leah with Headwaters Science Institute, and welcome to another Lunch with a Scientist. Today, you are going to hear about some brilliant research from Tanner Dulay. First, I want to give you a reminder of some of the things that are going on with Headwaters. So, primarily, we are preparing for our spring research experience for high schoolers. It runs February 2nd through April 23rd as an after-school extracurricular program, and students get to pair directly with professional scientists to create their own research project around almost anything that they're interested in, so they can totally personalize the program. It's great college prep as they emerge with a finished research presentation and they author a research paper that they submit for publication. So really good experience to set up a high schooler for college, for career, and just general critical thinking skills for life. So like I said, that program will be after school. There's 2 sessions, either Tuesdays at 4 or at 5:15 for the group meetings, and then the mentor meetings are one-on-one depending on your schedule. So register by January 22nd to get a spot in this program, and you can find more information on our website. So moving on, we're going to welcome Tanner Dulay in just a minute. Tanner is a PhD student at UCLA, and there he combines mathematical models and experiments to try to understand what makes ecosystems stable. So he previously studied the behavior and evolution of salamanders, and then he helped with a project on how climate change will affect plants and animals in California. So he became really fascinated along the way by how we can use math to create models that make sense of the way organisms interact in nature. So you'll hear all about that today, specifically in the context of the evolution of salamanders. He's going to talk about how ecologists have used clay models to explain what's going on with a common backyard salamander right before our very eyes. So without further ado, very pleased to welcome Tanner Dulay. Hi.
Hi, Leah. Nice to be here. Thank you for the introduction.
Absolutely. We're very grateful to have you. And before we jump in, I just wanted to ask you, I think this might be inspiring to some of our watchers. You are a PhD student at UCLA, but I understand that you're not talking to us from LA. Where are you coming to us from?
I'm currently sitting in my parents' kitchen in San Francisco. I have been working on my research remotely and teaching TAing remotely as well. And since that doesn't require me to be in Los Angeles, I've taken the time to be closer to family during the pandemic and try to work from home.
And can you tell us what does it mean to be a PhD student studying remotely during a pandemic?
Well, I think that's an interesting question that probably varies among what everybody studies. I think I'm very lucky because right now I'm working on theory. I'm working on mathematics behind how organisms interact. And so because of that, most of my research is computer based in the first place. So for me, anywhere that I could be with my computer kind of is the same as being in the lab for me. But for other people, I imagine it can be very difficult if they're away from the lab for any given reason, or if they have to stay in a campus that has very few people there and can't be near their families. I imagine it's a very challenging time, but yeah, I'm very lucky to be in the position that I am.
Great. Well, we're glad that you were able to adapt. And of course, everyone's using technology, and that's how we've got you here with us today. So we're very grateful for that and excited to hear about salamanders and all of your research. So without further ado, I'll pull in your presentation and let you take it away.
All right. Thank you so much, Leah. So good morning, everyone, or good afternoon, depending on the time zone that you're in. Today, I would like to talk about what I think is one of just the coolest stories in California, just natural stories, something that has just occurred in nature, and that is the story of the Ensatina salamander. So if you've ever gone outside and flipped some logs in your backyard, if you live in a wet-ish foresty area, you may have found a small gummy-looking lizard that isn't quite a lizard. They're amphibians, and these salamanders are really cool, and they're a lot cooler than I think a lot of people might originally expect when looking at them. They're pretty modest, and I think that a good science is all about telling a good story, and I think that these guys tell a fantastic story on their own, and I'm really excited to share it with you. So, ecology meets evolution, socially mediated speciation of the salamander Ensatina eschscholtzii. So this story begins here. in the super damp Pacific Northwest rainforests that stretch down into the very northwestern corner of California here on this map. 21 million years ago, this little salamander emerged. And if we were to walk around this forest and flip over some logs, we might find this. This is Ensatina eschscholtzii, or as we're just going to call them, Ensatina. So Ensatina, they look kind of like lizards, but they're not. Like I mentioned, they're amphibians. So they have a couple of very unique features that differentiate them from lizards and other animals. And one of them for the Ensatina is that it does not have lungs. It has no lungs at all. Instead, it has to breathe through its skin. And in order to do that, its skin has to stay very, very damp and moist. So they have to be in a wet environment. They're always underneath logs or in these very wet, mossy forests. And because of that dependency on being in a wet environment, the habitat that it can live in is very, very important. It can only live in very specific places, only places where there is adequate moisture for it to do this breathing through its skin. So it originated up here in that northwest corner of California, and from there it started to spread southward into California. But very quickly, it encountered a barrier that it could not pass, the Central Valley. If any of you have ever been to the Central Valley of California in the summertime, you know that it's a very, very hot and dry place. And at one time, it was even an ocean. The Pacific Ocean at one time filled the Central Valley. And whether it was full of water or completely dry and hot like it is now, the Central Valley is an inhospitable environment for these salamanders that need to stay moist to breathe through their skin. So what the salamanders did, what Ensatina did, is it stuck to the mountains on either side of the Central Valley, the Sierras in the east and the coastal range in the west. And in these mountains, you have these similar temperate forests where there are lots of Douglas fir and different kinds of coniferous trees, lots of moss and high levels of precipitation, lots of rain and even snow. And this wet environment was able to facilitate their breathing, and they could live in this environment and thrive there. And so they formed these two separate lineages. And the salamanders moved south in those mountains, staying there, and it took them a very long time, over millions of years. They slowly moved down the axis of the state until they came to the transverse ranges north of the Los Angeles area, and there they came back together. The mountains crossed the Central Valley area, and they were able to stay in this the wet, temperate forest environment up in the mountains and close this ring. So along the way, over these millions of years that they were moving south along these mountain ranges, they acquired a lot of random mutations in their genetic code. Everybody does. Anything that's alive and has DNA acquires just a bunch of random genetic mutations through your life. Some of them stick, some of them don't. Some of them you pass on to your offspring. And these random mutations occasionally make them look different and adapt to their local environments. So what happened is, as the salamander moved south, in these two different regions of the state, it adapted differently to those different regions. The ones in the east branch here adapted differently and look different from the ones in the west. So what do they look like now? Now we are left with this awesome ring of diversity. So we start up here with the Organensis and Picta, these two salamanders up here in the top left. These are closest to the ancestral Ensatina. And as they moved south in the coast range, you have this solid color crypsis going on over here. And in the Sierra, we have this blotchy pattern. So there are now 7 subspecies in total, these 7 subspecies that encircle the Central Valley. And they're subspecies, not different species. These can all interbreed. If any one of these salamanders bred with another one, if Platensis, for instance, up here on the top right, bred with Croceator, the yellow spotted one here. I see that my mouse is not on top of the presentation, so I'm just going to move that. If those two salamanders were to interbreed, they could have offspring. They would produce actual living, fertile offspring, which means they are the same species. However, at the very bottom of the ring, where eschscholtzii and clobberi, these two at the very bottom, here, oh, I can't even get to it, darn. These two at the very bottom, when they met, when this ring came down and closed, when the coastal branch met the Sierra branch in Southern California, those are no longer the same species. They have become so different as they moved down the axis of the state that when they interbreed, if they interbreed, they produce offspring that are sterile. They are not. They're kind of like a mule when a donkey and a horse has a baby. They're sterile. They're not a viable species on their own. Those offspring, and so we say that Schultzii and Clawberi have a species level divergence between them. They are so different that they are no longer the same species. However, Schultzii and Xanthoptica are the same species, and so is Picta and Organensis. All of them around the horseshoe make fertile offspring. So they are all the same species, except for at that joint. So that begs a really interesting question that people have been wondering about for decades now. How many species is this? Is this one species? Is it two? Is it seven? And generally, we accept that there's one species broken into these 7 subspecies, and that they are kind of on their way to possibly becoming more species. And that's going to be the focus of what I'd like to talk to you all about today. So, one notable feature of all of them, they all have this in common except for one. They're all cryptic. So cryptic is camouflage. All of these salamanders are camouflaged in their environment. I know that might seem a little counterintuitive because the ones on the right side over here are very brightly colored and spotty, but those spots actually work in a way to make them camouflaged. If you were to see one of these salamanders on the ground in their environment, those spots essentially break up their outline. So if a bird or a predator sees them, it doesn't see them, so to speak. It works the same way that camouflage that we think of with the big splotches on it kind of works. And the ones on the left are a little more obviously cryptic camouflage because they're dark. They hide in this very damp brown forest environment, and they're very effective at staying hidden in this environment. All except for Xanthoptica, this on the bottom left. So what is going on with Xanthoptica? It is not cryptic at all. This is what Xanthoptica looks like. Bright orange belly, bright yellow eyes. You can see it a mile away, super bright. So the question is, why, if all of the Ensatina are cryptic, is Xanthoptica not cryptic? And the answer is this. Xanthoptica lives in the same place as this newt, Pterica turosa. So this is called the California newt. The California newt is highly toxic. They have a very bright orange belly and bright yellow eyes. And what this does is it advertises to predators. It tells them, do not eat me or you will die. This is warning coloration, right? And so what happened is when Xanthoptica first came to the area where the newt lived, any salamanders that looked like the newt benefited from that warning coloration. If Xanthoptica has yellow eyes and an orange belly, predators will see that and go, ooh, I know what happened when my buddy Eric ate the bright orange and yellow newt. He died, right? And so I don't want to eat that, so I'm going to leave it. And so they are what we call a Batesian mimic. They're a mimic of the newt. And we call it Batesian mimicry because they are not toxic. Xanthoptica, the salamander, is not toxic at all, while the newt is very toxic. And this is a really interesting thing that happens in nature occasionally and has very interesting effects in the environment. Consider if there were a lot of these salamanders, if there were tons of them and very few newts, predators might start to realize, hey, not everything that looks like this makes me sick or kills me, right? And so I should start eating things that look like this because they're easy to see. So that makes a very interesting balance between these two, right? You need to have enough newts that the predators know that it's poisonous and not too many salamanders because they would kind of ruin the gimmick, so to speak. So they're a Batesian mimic. And this not only has an effect on the relationship between the Ensatina and this newt, but it also has a very, very important effect on how the salamanders interact with each other. So at one time, the Central Valley, I mentioned this is the inhospitable barrier, but at one time it was a little wetter. A few million years ago, it was wet enough that Xanthoptica, who lives in the Bay Area, was able to cross the Central Valley and reach the Sierras. The Central Valley then got dry again, as it is now, and it broke up that little bridge between the two, but we still have a population of Xanthoptica living at the base of the Sierra Nevada mountains. And right there, it meets with Platensis, which is the Sierra Nevada Ensatina. And these two are going to be the star of our show today. So Potensis and Xanthoptica meet where they shouldn't, right? The ring has been broken. We have this unbroken ring that goes all the way from the northwest down to the Southern California closure. And Xanthoptica, at this brief period, was able to jump across it. And so now we have this interesting dynamic where we're halfway down the divergence. At the top of the ring, the species are the same species. They can interbreed easily. Everything is fine and dandy. At the very bottom, they're totally different. They're not the same species anymore. They're too diverged. In the middle, we have this bizarre mid-ground where Xanthoptica and Potensis are pretty diverged. They have very different colorations, but they still hybridize, and they produce actual hybrid salamanders that are viable offspring. They are fertile themselves, and they can reproduce. So they are not technically different species. However, where they make the secondary contact zone, the hybrids only occupy a tiny little area. So you can think about having Xanthoptica here, Potensis here, they meet, they have babies in the middle, and when you have these hybrids, we used genetic assessments, or other researchers have used genetic assessments to find out that it is not good to be a hybrid in this environment. They exist, they can interbreed, they can do anything any of the other salamanders can do, but for some reason, they have lower fitness. So fitness in terms of evolution does not mean that we have like really jacked salamanders. What fitness means is the ability to produce offspring, the ability to reproduce and make baby salamanders that go and spread your genes off into the next generation, right? So these hybrids do that very much less often than either of the parents, Potensis or Xanthoptica. These hybrids suffer from significantly less fitness than Xanthoptica or Potensis, even though they come from those two. So the question we asked is why? Why is it bad to be a hybrid? And we think the answer is because when you have these hybrids form, you have two very, very different strategies of predator evasion that are coming together. On the left, Xanthoptica is brightly colored. It's a mimic. It wants to trick predators into thinking it's poisonous. The only way to do that is to be bright and loud with your colors, right? You want to be seen. Potensis, on the right side, has these spots that break up its outline. It wants to stay completely hidden. So what happens if we have them interbreed and then have a hybrid? We might have something that looks like this. Not really good at either strategy. They're not very brightly colored. They don't have the same bright yellow iris. They don't really look as scary. They don't really look like the newts that the Xanthoptica is trying to mimic, but they're also pretty orange, and they don't really hide as well in their environment as Potensis does. So we have this really interesting hybrid that isn't probably that good at hiding from predators because it has these mixed signals. And that is exactly what we think is causing them to have less fitness. Our hypothesis was that hybrids have lower fitness because their mixed phenotypes, the mixed colors they have, make them more susceptible to predation. They're not scary like Xanthoptica. They can't hide like Potensis. And we think predators pick them off more often because of that. So we have this hypothesis, and now we have to say, how are we going to test that? How can we go out and figure out if that's actually the case? And to do that, we threw it all the way back to preschool. Here are our three salamanders, Potensis in A, Xanthoptica B, and the hybrid in C. And we took clay, little Play-Doh, and built clay salamanders to look like each of the actual salamanders. And you might be a little confused by the hybrid there in F. The hybrid salamander model we made might not look exactly like the hybrid above, but we tried to capture as much of its important characteristics as we could. The bright orange on the back, the diffuse yellow eye, so it's almost like the yellow-eyed Ensatina, but not quite all the way there. And it was very important that we had the same amount of orange paint on each of these salamander models that we presented to the breeders. So what we did is we built all these models, spent hours painting salamander models in the lab, and then we took them to the actual hybrid zone, and we left them out in the sun for about 6 weeks, there it is. And we let them interact with the actual predators. We let the actual predators that would attack the real salamanders attack these models over the course of 6 weeks. And after 6 weeks, we went out and collected all the models, and we went and said, okay, that one was attacked by a predator. That one was attacked by a predator. And it was usually pretty obvious. Birds will peck them right on the back of the head. And then usually they go, that's clay. I don't want to eat that. But they leave a mark. The clay is very soft. It's Play-Doh. You've all used Play-Doh at some point, I'm sure. So this is where we deployed them. We deployed them in these transects where we randomized which type of model was next to which type of model to make sure there weren't any effects of having one area where it was all Xanthoptica or all hybrids. And this is the actual GPS we used to place and then find our models after 6 weeks. And what we found when we brought all the models back to the lab and we looked at who was damaged and who wasn't, we found that the hybrids were attacked way more often. Hybrids were attacked twice as often as either Potensis or Xanthoptica, more than twice as often. So what this tells us is that if you are a hybrid salamander, if you have those hybrid colors, you are more likely to get picked off by a predator and attacked. And if you get picked off by a predator, your fitness is done, right? If you die, if you're eaten, you're not going to have any babies after that. You're not going to produce any baby salamanders. So this wasn't. Oh, and this is what we call a disruptive selection gradient. This is just the relative survival of each type relative to one another. So you see the hybrids have this dip. Hybrids survive less often than the pure Potensis or pure Xanthoptica. So we have this amount of hybrids that are killed by these predators more often than Xanthoptica or Potensis. And we found that they were predators are enacting 19% selection against these hybrids. However, the studies before us found a lot more selection. The hybrids have even less fitness than can be explained by our clay models, basically. Our clay models showed that predators attack them this much. However, they do even worse than that in nature. So the next question is, why might that be the case? Why are we not showing the full picture here? What else is happening that is making the hybrids do so bad in their environment? And the answer we came to is that models, these clay models, just aren't the same as living salamanders. Living salamanders behave. When they encounter a predator, they do something, right? They don't just sit there like a stupid clay model. They actually do something. They interact. They use behaviors to amplify their predator avoidance strategies. So this leads us into part 2, behavioral divergence. New hypothesis. These behaviors that the real salamanders have are going to enhance the signals. So we think that for Xanthoptica, they're going to behave in a way that is consistent with them wanting to be seen, wanting to show off that warning coloration. And we believe that the Potensis and all of the other cryptic subspecies, the camouflaged ones, are going to behave in ways that enhance that make them look more camouflaged. So they're the two different strategies. Like I said, cryptics want to hide and stay hidden. Aposematism is a very difficult way of saying warning colors. And that's most effective when easily seen. And like I said, we think that our hypothesis is that the mimic species is going to behave differently than the cryptic ones in order to enhance these signals. And so in order to test this, we had to go find some salamanders in the wild. So we went all over the state, all over California, throughout the entire range of Ensatina, and we found every single subspecies of Ensatina in their natural habitat. The way we did this is we would go walk around the woods and flip over all the logs we could possibly see, and then, of course, very gently put them back. You don't want to disrupt the environment that the salamanders naturally live in when you're gone. And when we found a salamander, we'd observe them for 3 minutes. During that 3-minute period, we would probe them to simulate predation, and we would do that twice during the 3-minute period. And the probing looked like this. We just poke them on the head. You look at them really close and poke them very gently on the head, but just enough to simulate predation. What this does is it kind of scares the salamanders, and it makes them act as if they're encountering a predator. It makes them do what they would do if a bird just came by and pecked them on the head. And then we record what they do. What are the behaviors they do when they think they're being attacked? And we differentiate how we record these behaviors in expression versus latency. Expression is whether or not they do the behavior. Latency is how long it took them to do a behavior. So we compared all the subspecies, and here are some of the behaviors that we might see. there's this really characteristic behavior that some of the salamanders do when they think they're being attacked, where they stand up on their toes really tall and they arch their tail. And they make their tail super arched, and they even rock back and forth and wave the tail around like this. And other salamanders will run. You tap them on the head and they take off. They go running for little holes like this guy on the right, and they go try to hide. Or sometimes they just sit completely still. You poke them on the head and they just don't move at all, right? And that seems like something that would be a good thing to do if you wanted to stay hidden. If you think that your camouflage works, you don't want to move. You want to sit totally still and let nobody see that camouflage, right? So we recorded all these behaviors, recorded how long it took them to do it, and we did some very interesting statistics here. Here are a couple of salamanders in my hand. If you ever go outside, do look for the salamanders on your own, but if you're going to hold them or touch them, make sure you wash your hands very thoroughly before and after touching them, and preferably wear gloves if you're going to hold the salamanders, because not for your own safety so much as for their safety, because there are a lot of things on your hands, a lot of oils and salts and things that are not good for them. Remember, they breathe through their skin, and so they need to have that skin really clean and pure, and the things on our hands can be bad for them if they're not very clean. So we took these behaviors and we performed a multiple correspondence analysis. So before I show you what the actual data looks like here, what this graph is going to show us is anything that is closer together, any 2 points or subspecies that are closer together are more similar in their behaviors. The further apart two things are, the more different they are in how they behave. And so here is our data. So we have Xanthoptica, the hybrids, all of the other subspecies. Everybody is here on this multiple correspondence analysis. And what we can see really clearly is that all of the cryptic species, all the cryptic subspecies are in blue, and Xanthoptica is in green, and they're very, very different, right? They're super far apart on this graph. So they behave very, very differently. The amount of times that they do these behaviors are very different. And we can see that Xanthoptica on the left, it's clustered with some of the specific behaviors. It's clustered with the toe stand and the tail arch and the rocking especially. So it rocks and does its tail arch more often than all the cryptic species do. And this is, we call this a multivariate ANOVA, MANOVA, analysis of variance. And this is for the latency. How long does it take them to do these three behaviors, the toe stand, the tail arch, and the rocking behavior? So we found that in those 3 minutes, the majority of the subspecies up here on the top, it took them the whole 180 seconds. They just didn't do it. 180 means that they did not do the behavior at all. Whereas the Xanthoptica did it pretty quickly, right around 120, right at the first poke at 2 minutes. Right when we poke them, they do it. They get up on their tails, get up on their toes, and they rock that tail back and forth. And right in the middle, we have these hybrids. The hybrids take an intermediate amount of time to do this behavior, which is even more interesting, right? It's perfect. We have all the subspecies taking a long time to do this behavior. Xanthoptica does it really fast, and the hybrids do it at an intermediate time scale. It's perfect, right? It's just exactly what we would expect. So what we find is that Xanthoptica is quicker and more likely to do this behavior. It prefers to do this tail arch and toe stand and rocking behavior. But why? Why is it doing that in the first place? And the answer is they've taken an old trick and used it for a new purpose. All of the Ensatina, cryptic or mimetic, all of them can do this tail arch behavior like this. They all can do it. So it's not a specific Xanthoptica thing, but Xanthoptica does it so much more. And the reason being, they are newt mimics once again. So this is the newt on top. And when the newt is attacked, it wants to show that fiery underbelly to show predators, look at me, I'm poisonous, don't eat me. So they have this behavior called the Unken reflex. Unken reflex. When they get poked, when they get attacked, or when they think they're being attacked, they show their bellies like this. They pick their tail up, they pick their head up, and they get up on their toes a little bit. It's not exactly the same thing, but doesn't what the Ensatina on the bottom here is doing kind of resemble it? It's getting up on its toes, it's lifting that tail. It's a similar behavior in some ways, even though this is a totally different thing. The reason that the salamanders lift their tails is because they can detach that tail. So what they're doing is picking the tail up, hoping that a predator will bite the tail and they can run away when the predator is preoccupied with the detached tail, and their tail will then grow back. So this Ensatina, the salamander, is not trying to show predators that it's poisonous. It's not poisonous, remember? It's a mimic. But it still does this behavior more often because predators have attacked them less often if they do it. If you're a predator and you see somebody that looks like this, bright yellow eyes, bright orange belly, and you give it a peck just to test it, if it puts its tail up, then you go, yeah, that's definitely this dude that killed Eric, the other day. I'm not going to eat that. If you see the bright yellow eyes and the orange belly, and you peck it, and it doesn't do that behavior, if it didn't raise its tail, then you might say, aha, I found a cheater. This one's not poisonous. I know that. I can eat this. And so if you don't raise your tail, you have lower fitness because you get picked off by predators more often. So because of that, this behavior of lifting the tails has been selected for in the Ensatina, and so they do it way more often. So now what we have is Xanthoptica and Platensis not only look differently with these different colors and patterns of evasion, they also behave very differently. And so now the question is, why does that matter? Why does all this matter? What does it mean if they're diverging in two different respects? They're behaviorally diverging and they're morphologically diverging. And this leads us into part 3, which I think is pretty exciting. And that is that salamanders play rock, paper, scissors. That may sound pretty silly, but I promise it is real, and I'm going to talk to you about it right now. So these 3 guys play rock, paper, scissors. So in the hybrid zone, we have all 3 players in one place. We have Xanthoptica, we have Platensis, and we have the poisonous newts, all of them coexisting in the hybrid zone. Most of the places where we have these, most of the intergrade zones here, so if we look up between any other 2 colors, if you look between Platensis and the blue one, right, or the blue one and the green one, all of the color changes are actually pretty gradual. Most of the cryptic species, they have a long range of intergrade zones. And if you were to actually go to all these places, you would find that the change in color from one place to the next is pretty gradual. It's pretty slow. They're all pretty similar, except for here. Xanthoptica to Platensis is sudden. There's a break, right? You have bright colors and you have camouflage right next to each other. And that may seem trivial, but it's actually a very complex issue. Coexistence is surprisingly difficult to maintain in nature. People work on this a lot. People don't quite understand, even today, how different things can coexist. And the reason why doesn't seem very simple at first, but it really is. So this is a little computer simulation that I ran of this red up here is a resource. It's like a plant that 3 species are sharing. Okay, so we have the blue, green, and yellow species here on the bottom. These are like herbivores that are eating this plant. And this is how many of those species are around over time. So what we find is that if C1, this first consumer, the blue one, if the consumer is the better competitor for this resource, it drives the other 2 to extinction, always. No matter what, if there is any kind of imbalance, unless they are perfectly the same when it comes to competing for this resource, only the best will survive, right? Only the survival of the fittest, you might be tempted to say. However, there are some things that can subvert this. So rock, paper, scissors, what we call intransitivity is the mathematical term for it. Rock, paper, scissors allows them to coexist. So here's another simulation I did of the exact same thing where now consumer 1 beats consumer 2, consumer 2 beats consumer 3, but consumer 3 beats consumer 1 again. Just like rock, paper, scissors, right? 1 beats 2, 2 beats 3, 3 beats 1. You could literally play rock, paper, scissors with those numbers and it would work. And what happens is now they do this thing where look at them each kind of fluttering like that. When one of them becomes really common, when C1 is really common, it's really good to be C3, right? Because C3 beats C1. So then C3 becomes common. And they do this thing where they trade off who is the most common species over time, and they can all coexist like this forever. And we see a very similar dynamic happening with these salamanders. So why do we have Xanthoptica and Platensis and this newt all coexisting when they all basically have the same niche? They have the same resources, they have the same environment, same habitat, and yet they can all coexist and not outcompete one another. And we think that's because they're playing rock, paper, scissors. And we call this special game of rock, paper, scissors A, B, C, and R. So I'm going to run you through what each of those means. A is the aposematic model. The poisonous newt is A. Okay. B is the Batesian mimic, Xanthoptica. Bright yellow eyes, bright orange belly, not poisonous. Batesian mimic is B. C is the cryptic prey, the Platensis here, who has the camouflage spots on his back. And then the N and R refer to the predators here. So here's they're one of their very common predators, the Steller's jay. It's a kind of a noisy bluebird that'll pick the heck out of these guys, and it loves to eat salamanders. And these predators can either be naive, meaning they do not respond to the signals if they come up. They see the warning colors and they don't care. They don't know any better. Or they could be responsive. They see the warning colors and go, I know that one. I know this one. I'm not going to eat it, right? And so what happens is when the predators interact with the salamanders, they create a rock, paper, scissors game. If you are the aposematic model, if you're A, then it's good. No, it's good to be A. But if you have this bad, if you're poisonous and have this warning coloration, and you're really common, then it's good to be B. Because B gets the same benefit, or predators will leave it alone, but it doesn't have to invest in actually producing the poison. So if you have A, B wins. If A is very common, B will then take over and become even more common, right? Once B is really, really common, then the predators will go, hey, wait a second. I know this. And it's really easy to find these guys, and they're not always poisonous. In fact, there's so many of them now that I'm not even afraid to eat them because the risk of getting sick is so low. So then predators will start eating B, because they are so common and so easy to see, right? They have this bright orange and yellow coloration, and it's really hard to find C. C is camouflaged in its environment. So if the predators, the birds, have this really easy to find prey in B, then why would they bother trying to find the cryptics, right? So if B is super common, then it's good to be C. C beats B. Right? So the Platensis here, the cryptic on the bottom right, becomes very common now when B was common. It does better in that environment. And then once the cryptics are very common, then it's good to be A again, right? Because now cryptics are so common that the predators might say, okay, well, there's so many of these guys that are never poisonous that why should I risk attacking something that looks like it could make me sick, right? So then once C is very common, it's good to be A again. So the predators themselves are making these salamanders and the newt play this rock, paper, scissors game. And there are some very, very interesting implications of that. They play rock, paper, scissors. And what we do is we convert this dynamic into equations. Oh, shoot. Sorry. Whoa. I made that slide twice. That's my bad. So we take those dynamics and we can form it into an equation like this. So this big triangular prism here, what this shows us is the space where they can coexist. This is called a simplex. And if you imagine a dot, a point anywhere inside of this prism, that point represents what the proportions are of A, B, C, and N and R in a population at a given time. And so we can say, starting in any place inside this prism, what's the outcome? Where will it go to? Where does this population tend to if we leave it and let it go with this dynamic? So we can model this using computer simulations of these equations. And one thing to note is that if you start inside the prism, that means everybody coexists, right? You have A, you have B, you have C, and N and R. If it moves onto a face, for instance, if it moves up to this face on the top right that's formed by A, C, N, and R, if it goes onto that face, that means B has gone extinct. There's no more B. And once B is gone, B can't come back. So what we want to ask is when we use these equations and see what happens, we want to know what are the outcomes? When we have all these things coexisting, what happens to them over time? And what we find is that in these hybrid zones, where you have A, B, C, and NR all coexisting, the model shows us that there are interior equilibrium. So that means there's a point that is stable inside this prism. And when there's a point that's stable inside the prism, not on one of the faces, that means, again, that everybody coexists. Everybody can be there, right? And they can coexist together and do this thing where they don't outcompete one another because the predators are making them play this game. So the model tells us that, yes, this is mathematically stable. These communities can coexist, but only if B and C are very distinct strategies, right? We have to have the Batesian mimic being a Batesian mimic, and we have to have a cryptic being a cryptic in these environments. And so the final thing to leave you for here is that those predators, by making them play this coexistence game, the coexistence game is only stable if B and C are divergent. So they have these divergent behaviors, these divergent colorations between Platensis and Xanthoptica. And that is because these predators are forcing them to be separate species entirely. So the specific anti-predator behaviors enhance those signals. Hybrids have this mixed phenotype that's bad, so they get picked off more often. Predators will enact selection. They give higher fitness to pure Platensis and pure Xanthoptica because of these signals. And what this does in the long run is, if it's bad to be a hybrid, after a long time, you reach a system where if you are Platensis, then you say, I don't think I want to mate Xanthoptica anymore, because if I mate with Xanthoptica, then my hybrid offspring are going to get eaten by predators. If I am Platensis and I just mate with Platensis, then I get Platensis babies and they can hide and they do very well in the environment, right? So it's very good to mate within your species if this dynamic occurs. And that is exactly what we see. We see that, we see this dynamic starting, that the predators are forcing these two salamanders to become different species right before our eyes. So right now, they're still the same species, but they are on their way to becoming new species. So next time you go outside and flip over some logs and find a cool salamander, you can know that as simple as they are, they represent pretty much every single aspect of evolution and ecology that you can possibly imagine. And they are just incredible, super cool little gummy lizards, and they tell a really beautiful story. So at the end of the talk, I'd like to throw out some acknowledgments. I'd like to thank Professor Barry Cinervo at UC Santa Cruz for conceiving of and helping us with all this work. Everybody who helped me in the field and in the lab. And of course, the Headwaters Science Institute for providing the platform for this talk. This has been a lot of fun, and I look forward to hearing any questions that you have.
That's fantastic, Tanner. Thank you so much.
Yeah, no problem. Thank you for having me.
Absolutely. I had no idea that the fate of one salamander was so dependent on the entire species. Pretty interesting. So we've had a bunch of watchers here, and I just wanted to point out Morgan here, who is listening while gardening and found a salamander while you were talking. So I thought that was pretty interesting.
Oh, I know Morgan. That's awesome. Great.
Yeah, we're talking about something in everyone's backyard. So it's great. So I'm going to open it up here for questions. And I just wanted to ask a kind of general question to kick us off about how you got into exactly where you are with your educational path and career. So I noticed that you became interested in ecology, you became interested in salamanders, but how did that turn into really wanting to use mathematical representations of data specifically?
That's a good question. So when I first went to college at UC Santa Cruz, I originally started as an astrophysics major. So I was really interested in physics of stars and galaxies and how everything out there interacts. And that's, of course, very mathematical. And I was studying this, and I just didn't feel very close to the research. It all felt very far away and not really. A friend of mine once put it very, very simply that space is really cool, but you can't really do anything about it. And to me, that was a kind of a prophetic way of saying that I should look for something that I can interact with, because I'd feel more engaged. And so that kind of led me to start studying biology during college. But halfway through, I switched to biology. And along the way, I did a field course that was fantastic. It's called California California Ecology and Conservation field course. And it was a great experience, my first real exposure to doing hands-on research. And I think that using math to explain these really complex systems is a beautiful thing, right? We can take this incredibly messy and really beautiful system that we find in nature. They're really not very well organized, and derive equations to try to understand why they exist, to make sense of all the craziness that's out there. And I think that it's just a really beautiful thing that we can do that in the first place. And a lot of ecological theory has lent itself to really great conservation. And I just think it's a lot of fun. It's really beautiful.
Yeah, it's always fascinating hearing kind of how you got to your specific place in your career. It's quite an interesting journey. I'm sure some of the students who will be watching this talk will be fascinated about how math and salamanders relate to one another. So let's get to a few questions here. This question is from Morgan. With the relatively new evidence of California native beavers and their water management along the Sierras, do you have any information about how beaver dams and ponds have helped salamanders jump the Central Valley, if they have?
That is a really, really good question, Morgan. I'm not familiar with the interaction between the beaver ponds and the salamanders themselves. That's a really, I would expect there was something, suspect there was something there, because I know that in other systems, like in Yellowstone, the presence of beavers building dams and rivers pretty much provides the environment that other amphibians need, like certain toads and frogs that live in Yellowstone, because they slow down the rivers and create these places where it's no longer so fast moving that frogs can't lay their eggs there. So I wouldn't be surprised if that exact same kind of dynamic occurred in the Central Valley and facilitated the jump across the Central Valley that the Xanthoptica made. That's a really fascinating question. I would love to look into that more. That's awesome. Thank you.
Yeah, I'm sure we're all kind of dying to know, how did they get there, Okay, so this one's kind of about the breeding. Any idea on what the hybrid breeding returns for the Western Sierras? If we see species one searching out species one for breeding, then do the hybrids tend towards species one or species two?
Ooh, another very good question. Maybe I wasn't clear about this in the presentation. So that we have not actually done an experiment to see if there is this assortative mating yet. So we don't actually know if species one is seeking species one specifically and species two is seeking species two specifically. Right now, we see just a lot of hybrids with very low fitness in this very narrow range. And I don't think anyone has even started to look at who they breed with, because their breeding is very poorly understood. I'm pretty sure they mate underground. And I think the first videos actually of Ensatina's mating just came out like in the last 5-10 years, I think. So, or maybe even more recently than that. But we don't really know. That's a really, really interesting question. I would love to know the answer.
You've got a lot of ideas here for your future research, definitely.
Yeah.
This one's coming from Lara, one of our summer research program students. Hi, Lara, how are you? So regarding the toxin on salamanders, do all of them have some trace amounts? And how toxic are our backyard salamanders?
That is a great question, Lara. Thank you so much for asking. So in this case, the Ensatina, they don't produce any actual toxins, but it is, if you touch them, if you go out in your backyard and find them and then safely handle them, you should always wash your hands, because touching wild amphibians can make you sick because they have certain bacteria on them. But it's not a toxin. It's the same thing as if you were to touch like raw meat. You can get like salmonella poisoning. The newts, on the other hand, the Pacific newts, they produce a very, very powerful toxin called tetrodotoxin. And tetrodotoxin is extremely toxic, and you can see it if they're very agitated when we do the little poke to simulate predation. They sometimes produce the toxin in little glands on their neck, and it comes out in this sticky white substance. And the salamanders, actually, the Ensatina, they do produce another white excretion on their tails. But what they've found that's really interesting is that when the salamanders think they're being attacked, they produce this secretion that looks kind of like the poison, but it's not poisonous. it's really sticky instead. And so what they think this does is that this sticky substance they produce makes it so that if a predator attacks them and eats that tail, it's so sticky and gummy that it makes it so their mouth gets gummed shut so they can't attack the salamander afterward. So kind of crazy, but they're not really toxic, but they still do this secretion behavior that helps them. So awesome question. Really, really interesting stuff.
Yeah, that sticky substance sounds like another case for study as well. I'm curious, when you're out there looking for these salamanders, what does being a salamander hunter really look like?
Oh, it's a lot of fun. It's really just tromping through the woods and flipping over logs and looking for them. And sometimes it's very rewarding and it's easy. Some places they're just so abundant that they're everywhere, and sometimes they're pretty rare. That one of the subspecies, Croceator, that lives in the transverse ranges, it's listed as endangered in California because they live in a habitat that's really susceptible to private land encroachment, and so their habitat gets destroyed pretty frequently. And because of that, there's a lot of concern about how long they will be around for. But when we went out all over the state to look for these salamanders, there were some trips we went on where we spent a lot of money on gas to get across the state and then came up dry. We looked everywhere, flipped every log we could find for miles, and just didn't find any salamanders. And sometimes there was a majorly delayed gratification. One of the best ones was looking for picta that went up in the very far northwest corner of the state. We drove from Santa Cruz all the way up there. It took us like 2 days to get all the way to the top of the state. And we were looking everywhere and couldn't find them. Every log we flipped for a whole day, we found nothing in all these beautiful redwood forests. And it was a fantastic place to be out and exploring. But we were getting really nervous because we had spent a lot of lab money on gas to get all the way up there. And we didn't want to come back empty handed and have to tell our PI that we wasted all that funding. And we kind of took a gamble and drove an hour further north, expecting to get home in the dark. And we got super lucky. The very first log we flipped over up near Crescent City had 2 salamanders underneath it, and they were under every single log up there. Like, if you ever want to see salamanders, go to Crescent City. They're everywhere. It's insane. So that was really exciting. There was a lot of cheering and hooping and hollering in the woods.
Yeah, that's great. Glad you found them. So we're going to do a couple more questions here before we sign off. I know that you talked about kind of what you're studying, them being on their way to becoming a new species, but not quite. On the other end, have you ever helped to discover a new species?
Oh, hey, Anu, what's going on? Great question. Nope, I have not discovered or been involved in the discovery of a new species. However, they do discover new species of salamander pretty often. They're very, very ancient organisms that still live among us. And because of these really weird ways, they don't move very far. I think in Ensatina, they did a study where they found what the average distance an Ensatina moves in its entire life is. And it was something like 15 feet. They have like a 15-foot diameter around whatever their log is. And that's their home. They don't really go beyond that. And so because they move so little, you can have these dynamics where they don't interact with each other, even though they're very closely related. And you can have this thing called cryptic speciation. So you can have new species occur that can't interbreed, but you can't tell because they look the same, basically. There's not a lot of selection acting on them in some of these places. And so they just diverge kind of ambiently, genetically, because they're not interacting. So I think up around Shasta Lake, actually, there's a whole entire different species of salamander called the Shasta salamander. And just in the last couple of years, they did some genetic assessments and found that one species of salamander is actually, I think, 3 or 4 species of salamander. So no, I have not ever been involved myself, but it's happening all the time. Oh, and I think I want to say it was in China, but it might have been in a place further southeast in Asia. They discovered a brand new species of salamander that's very closely related to the Ensatina, but there's nothing else like them in that part of the world. And it's bringing up a lot of really interesting questions about how did they get here? How did they get there? Why do we have some of them over here and a few of them over there? And salamanders have these very bizarre distributions as, they just need these very rare special habitats. And
Mm.
when they die, they're so soft and squishy, they don't really leave fossils behind very often. And so it's very hard to find out where they used to live ancestrally.
Interesting. Really fascinating that we're starting to see them all over the place. We're going to wrap up with what I think is a very fitting question to kind of seal in everything cool we've heard from you today. What is the coolest salamander you've ever seen?
Oh, that's a great question. This is my sister actually asking the question, so I know that she knows the answer. One of my favorite animals ever, one of the coolest salamanders, besides the Ensatina. I think the Ensatina are incredible just because of the story they tell. I mean, you get every single aspect of evolution in one beautiful little gummy lizard. It's just so cool. But up in the very high Sierra, there's a species called the Mount Lyell salamander. And the Mount Lyell salamander is incredible because they live in an even more extreme environment. They're adapted to living only on vertical granite faces, like mountainsides, that have to be below a glacier or permanent snow that trickles down the wall all through the summertime. So they live on these walls all summer long. They just walk around. They have special adaptations to climb vertical rock. They're like Alex Honnold and Free Solo. They're nuts. They're super good climbers. And they just eat little bugs and insects that land on the wall in the summertime. And in the winter, these areas get up almost like 10 feet of snow or more in a good winter. And so they just dig down into the talus piles. They hide really deep in these mountains, and they go into this kind of quasi hibernation state. And it's just incredible to me. How can they have adapted to this ridiculously harsh and challenging environment? I think it's just amazing. And yeah, they're probably the coolest animal maybe in all of California, I think.
Well, there you have it. All different kinds of salamanders in all different climates, even in the snow. Really neat. Thank you, Tanner, so much for sharing your research. It was very vibrant, very alive, and really something that we can find in our backyard as well. So really appreciate it.
Yeah, thank you so much for having me. It's been a pleasure.
All right, we'll talk to you soon.
All right. Bye, Leah. Bye, everyone.
Bye.